This session on acquired immune response focuses on antigen recognition and activation. This topic is fundamental to understanding how our immune system specifically recognizes and remembers pathogens. By the end of this session, you'll be able to define acquired immune response and understand how it differs from innate immunity in its specificity and memory capabilities. Differentiate between primary and secondary immune responses. Describe how antigen recognition occurs and how this recognition activates immune cells in the acquired immune response, and various stimulatory, co-stimulatory, and accessory molecules involved in antigen recognition. Also learn to classify cytokines into those of innate versus adaptive immunity. And finally, you'll list clinical applications of cytokines, understanding how this knowledge translates into therapeutic interventions for infections, cancers, and immunological disorders. The acquired immune response is a specific immune response, meaning it targets particular pathogens with precision. Unlike innate immunity, which responds generally to broad categories of threats, acquired immunity recognizes specific molecular structures on individual pathogens. In this response, the immune system actively participates through lymphocytes, B cells, and T cells that undergo activation, proliferation, and differentiation. These cells don't just respond passively, but undergo complex changes that take days to develop. The primary function is to defend the host against pathogens through two main mechanisms: antibody-mediated immunity involving B cells producing specific antibodies, and cell-mediated immunity involving T cells directly attacking infected cells or coordinating immune responses. The key distinguishing features of acquired immunity include specificity, recognizing and responding to specific antigens; diversity, capable of recognizing millions of different antigens through gene rearrangement; memory, maintaining long-lived cells that enable faster responses upon re-exposure; and self-limitation, responses that eventually contract once the pathogen is eliminated. This session will explore how these remarkable properties emerge from antigen recognition and cellular activation processes. Here is a case illustrating primary and secondary immune responses. Sarah, a 25-year-old, contracts chickenpox after exposure at her new daycare job. This is her first encounter with the varicella-zoster virus. Following a week-long incubation period during which the virus replicates but symptoms haven't yet appeared, Sarah develops initial symptoms, including mild fever and fatigue. These constitutional symptoms reflect the developing immune response with cytokine release. She then breaks out in the characteristic itchy rash of chickenpox that has fluid-filled vesicles appearing in crops over several days. Over the next week, her symptoms peak as both viral replication and immune responses reach maximum intensity. Her symptoms then gradually subside as her immune system overcomes the virus through both antibody production and cytotoxic T-cell responses. 5 years later, Sarah is re-exposed to the chickenpox virus from her niece, who has active infection. This time, the outcome is dramatically different. Due to her previous infection, her immune system possesses memory B cells and memory T cells, specific for varicella-zoster virus. These memory cells quickly recognize and clear the virus. Sarah experiences only very mild symptoms, perhaps slight fatigue but no rash, and achieves rapid recovery without developing full-blown chickenpox. This is classic secondary immune response. This case perfectly illustrates the principle of immunological memory underlying vaccination strategies. The primary immune response occurs during first exposure to a foreign antigen. Understanding its characteristics helps explain why initial infections are often more severe, and why vaccination requires time to provide protection. Upon first encounter, the immune response is mediated by B cells producing antibodies and T cells, providing either help for B cells through CD4 T cells or direct cytotoxicity through CD8 T cells. These lymphocytes must recognize the antigen, become activated, proliferate, and differentiate. These processes take considerable time. The response produces high-affinity, antigen-specific antibodies or cytotoxic cells through a selection process called affinity maturation. B cells with receptors that bind most strongly to the antigen are preferentially selected to survive and proliferate. The first antibody produced is mainly IgM, a large pentameric antibody particularly effective at agglutination and complement activation. IgM appears first because B cells don't initially undergo class switching to other antibody types. The entire primary response can take up to 14 days to reach peak effectiveness. This lag period explains why people get sick during first infections. During this time, the pathogen replicates faster than the immune response develops initially. Critically, the primary response generates memory cells with high specificity for the exposed antigen. These long-lived memory B cells and memory T cells persist for years or decades, ready to respond rapidly upon re-exposure. The secondary immune response occurs following subsequent encounters with the same antigen. The dramatic differences from primary responses underlie vaccination effectiveness and natural immunity. Memory cells generated during primary response are already present and don't need to undergo initial activation and selection processes. Upon re-encountering the antigen, these memory cells rapidly activate and respond. A very short period is required for effective response, typically just 3 or 4 days, compared to 14 days for primary response. This speed difference is due to memory cells being more abundant, already having undergone affinity maturation, and requiring less stringent activation signals than naive cells. A much larger amount of antibody is produced compared to primary responses. Memory B cells can quickly differentiate into plasma cells, producing massive quantities of antibodies, overwhelming the pathogen before it establishes infection. Antibody levels tend to remain high for longer periods. While primary response antibodies eventually decline, secondary responses maintain elevated antibody levels for extended periods. The main antibody type produced is IgG rather than IgM. Memory B cells have undergone class switching, producing IgG subtypes that provide better tissue penetration, cross the placenta, and have longer half-lives than IgM. This explains why Sarah didn't develop chickenpox symptoms during her second exposure, and why vaccines provide long-lasting protection. This graph illustrates the quantitative differences between primary and secondary immune responses over time. The x-axis represents time after antigen exposure, while the y-axis shows antibody levels. Following first antigen exposure, indicated by the first arrow, there's a lag phase of several days where antibody levels remain low as naive B cells undergo activation and proliferation. Antibody levels then rise gradually, reaching a modest peak around 14 days post-exposure. The curve shows primarily IgM antibodies, represented by the lower portion of the curve. IgM appears first and peaks earlier. Some IgG production occurs but remains relatively low, shown by the upper portion. After the peak, antibody levels decline as short-lived plasma cells die and antigen is cleared. However, a baseline level persists from long-lived plasma cells, and memory cells remain even when antibodies become undetectable. Upon second antigen exposure, shown by the second arrow, the response is dramatically different. There's minimal lag time. Memory cells respond within 1 to 2 days. Antibody levels rise much more rapidly and reach much higher peaks than primary responses. The predominant antibody is now IgG rather than IgM, reflecting class switching. The response is also more sustained, with antibody levels remaining elevated longer before declining. Antigen recognition initiates a cascade of cellular events leading to effective immune responses. Understanding this process reveals how lymphocytes transform from resting cells into effector cells. The process involves 4 key stages. First is activation, where lymphocytes receive signals through their antigen receptors and co-stimulatory molecules, transitioning from quiescent phase into active cell cycle. Second is differentiation, where activated lymphocytes commit to specific functional fates. T cells may differentiate into various subsets including Th1, Th2, or cytotoxic T cells, depending on signals received. B cells differentiate into antibody-secreting plasma cells, or memory cells. Third is maturation, involving functional and phenotypic changes that produce fully competent effector cells. For B cells, this includes affinity maturation through somatic hypermutation and selection. Fourth are effector mechanisms, the actual functional responses that eliminate pathogens. For B cells, this means antibody secretion. For CD8 T cells, this means cytotoxicity. For CD4 T cells, this means cytokine production coordinating other immune cells. Lymphocytes are the key cellular mediators of acquired immunity, divided into 2 main lineages with distinct but complementary functions. B cells are responsible for humoral immunity through antibody production. Upon activation, B cells differentiate into 2 populations. Plasma cells are short-lived but highly active antibody factories, secreting thousands of antibody molecules per second. Memory B cells are long-lived cells maintaining immunological memory for rapid secondary responses. T cells mediate cellular immunity and come in several functionally distinct subsets. Helper T cells or Th cells provide assistance to other immune cells through cytokine secretion. Th1 cells promote inflammatory cellular immune responses and activate macrophages through cytokines like IFN-gamma and TNF-alpha. Th2 cells induce humoral responses by helping B cells through cytokines like IL-4, IL-5, and IL-6. Cytotoxic T cells or TC cells directly kill infected or cancerous cells by recognizing antigens presented on MHC class I molecules. Suppressor T cells or TS cells regulate immune responses, preventing excessive reactions. These are now understood as regulatory T cells or TREGs. T cells mediating delayed type hypersensitivity or TDTH orchestrate inflammatory responses at sites of chronic infection. T cells possess sophisticated molecular machinery for antigen recognition and activation. Understanding these components is essential for grasping how T cells function. The T cell receptor, or TCR, is the antigen recognition structure, consisting of alpha and beta chains forming a heterodimer. Each T cell expresses a unique TCR, specific for a particular peptide MHC complex, through genetic recombination during T cell development. The TCR complex includes associated signaling molecules, particularly CD3, which doesn't bind antigen but transduces signals into the cell when the TCR engages antigen. CD3 consists of several chains gamma, delta, epsilon, and zeta. T-cell co-receptors include CD4 and CD8 molecules that enhance TCR signaling and determine which MHC molecules T cells recognize. CD4 binds MHC class II molecules, making CD4 T cells responsive to antigens presented by professional APCs. CD8 binds MHC class I molecules, allowing CD8 T cells to recognize antigens from any nucleated cell. Cell adhesion molecules stabilize interactions between T cells and APCs, bringing receptors and ligands into close proximity. These include LFA1, ICAM molecules, and others. Activation requires integration of signals from all these components. Following successful activation, T cells undergo differentiation into various effector subsets depending on the cytokine environment, which we'll explore next. T cell development in the thymus is a remarkable process, producing a diverse repertoire of T cells that recognize foreign antigens while avoiding self-reactivity. Understanding thymic selection explains how the immune system achieves self-tolerance. Immature thymocytes enter the thymus lacking TCR, CD3, CD4, and CD8 molecules, designated as double-negative cells. These cells begin TCR gene rearrangement, a process similar to antibody gene rearrangement, creating enormous diversity. Successful TCR rearrangement produces double-positive thymocytes expressing TCR, CD3, both CD4 and CD8 simultaneously. These cells now undergo selection processes, determining which cells survive. The critical concept is MHC reactivity testing. Thymocytes are tested against self-MHC molecules presented by thymic epithelial cells. 3 outcomes occur based on TCR-MHC interaction strength. Cells with weak reactivity to self-MHC molecules survive and continue development. These cells can recognize self-MHC presenting foreign peptides, the desired outcome called positive selection. Cells showing no reaction to self-MHC die by neglect. These cells couldn't function as mature T cells, since they can't recognize MHC molecules presenting antigens. Cells showing strong reactivity to self-MHC with self-peptides undergo apoptosis through negative selection. These potentially autoreactive cells are eliminated, preventing autoimmunity. Surviving cells lose either CD4 or CD8, becoming single-positive mature T cells that exit to the periphery. This process ensures T cells are MHC-restricted and self-tolerant. T cell activation requires multiple molecular interactions, ensuring responses occur only when truly needed. This multi-signal requirement prevents inappropriate activation that could cause autoimmunity. Stimulatory signals provide the primary antigen-specific signal. This involves MHC molecules presenting antigen peptides recognized by the T cell receptor. This is signal absolutely required but not sufficient alone for full activation. Co-stimulatory signals provide the critical second signal, determining whether activation proceeds or tolerance develops. The most important is CD28 on T cells binding to B7 molecules, also called CD80 and CD86, on antigen-presenting cells. Without this co-stimulation, T cells receiving only TCR signals become anergic or unresponsive. Accessory molecules strengthen and stabilize T cell APC interactions, bringing cells into close contact and organizing signaling molecules. These include CD2 on T cells binding LFA3 on APCs, LFA1 on T cells binding ICAM1 on APCs, CD45R interacting with CD22, and additional CD28B7 interactions. The requirement for multiple signals makes biological sense. Signal 1 alone, MHC antigen TCR interaction, could occur with self-antigens, potentially causing autoimmunity. Requiring co-stimulation ensures activation occurs only when APCs are properly activated by infection or inflammation, indicated by upregulation of B7 molecules. This two-signal model is fundamental to understanding immune regulation and has therapeutic implications for preventing transplant rejection and treating autoimmune diseases. T-cell activation follows a carefully orchestrated sequence of molecular events, resulting in proliferation and differentiation. Understanding these pathways explains how diverse T-cell responses emerge. Signal 1 involves the primary interaction of MHC plus antigen with the TCR-CD3 complex. This provides antigen specificity and initiates signaling through CD3-ITAM phosphorylation. Signal 2 involves CD28 on the T-cell binding B7 on the APC. This co-stimulation is essential for full activation and prevents anergy. The result of receiving both signals is signal transduction, activating multiple intracellular pathways. The cell resumes the cell cycle, transitioning from quiescent G0 to active G1 phase. Many genes are activated, especially IL-2 and its receptor IL-2R. IL-2 is crucial. It's an autocrine growth factor, meaning T cells produce it and respond to it themselves. IL-2 binding to its receptor drives proliferation and differentiation, causing clonal expansion of antigen-specific T cells. The outcome yields different T cell subsets, depending on additional signals. Th1 cells are preferentially activated by IL-2 and IL-12 from macrophages and dendritic cells. They produce cytokines, including IFN-gamma and TNF-alpha, involved in activating inflammatory responses and cell-mediated immunity, particularly important for intracellular pathogens. Th2 cells are preferentially activated by IL-4. They produce cytokines including IL-4, IL-5, and IL-6, involved in activating antibody-mediated immune responses, particularly important for extracellular pathogens. This bifurcation determines whether immune responses are primarily cellular or humoral. B cells recognize antigens through the B cell receptor complex, structurally and functionally distinct from T cell receptors. Understanding BCR structure explains how B cells initiate antibody responses. The core of the BCR is membrane immunoglobulin, or MID, which is essentially an antibody molecule anchored in the cell membrane. Each B cell expresses a unique MID through genetic recombination, providing antigen specificity. The MIG consists of 2 heavy chains and 2 light chains, forming the characteristic Y-shaped structure with 2 identical antigen-binding sites. However, the MIG cytoplasmic tails are too short to transduce signals effectively. Therefore, the BCR includes associated signaling molecules CD79A. An important distinction is that BCRs recognize T-independent antigens that can activate B cells without T cell help. These are typically repetitive structures like polysaccharides that cross-link multiple BCRs simultaneously, providing strong enough signals for activation. However, for most protein antigens classified as T-dependent antigens, B cells require help from CD4 T cells. The B cell internalizes antigen bound to its BCR, processes it, and presents peptides on MHC class II to helper T cells, which then provide co-stimulatory signals and cytokines, enabling full B cell activation and antibody production. Cytokines are crucial signaling molecules coordinating immune responses. They are extracellular factors that affect cell proliferation and differentiation. Lymphokines are a subset of cytokines, specifically referring to proteins produced by T cells. Historically, this term was used before the broader category cytokine became standard. Lymphokines have effects on differentiation, proliferation, and activation of various immune cells. Cytokines serve multiple critical functions. First, they recruit circulating leukocytes and lymphocytes to sites of immunological encounter, functioning as chemoattractants that direct cell migration to where they're needed. They stimulate development and proliferation of B cells and T cells, acting as growth factors that expand antigen-specific clones. They stimulate and prepare macrophages for their phagocytic tasks, a process called macrophage activation that enhances their antimicrobial capabilities. They stimulate natural killer or NK cells, enhancing their cytotoxic activity against infected or cancerous cells. They provide antiviral protection, particularly interferons that induce antiviral states in cells, inhibiting viral replication. Cytokines act through specific receptors on target cells, often in cascades, where one cytokine induces production of others, creating complex regulatory networks. Let's examine specific cytokines of innate immunity, understanding their sources, targets, and functions. These cytokines form the first wave of immune signaling. IL-1 is produced by activated macrophages and acts as a growth regulator of T cells and B cells, promoting their proliferation. It's chemotactic for neutrophils, recruiting them to infection sites. Importantly, IL-1 is an endogenous pyrogen, meaning it induces fever by acting on the hypothalamus. This explains why infections cause fever. IL-12 is produced by macrophages and dendritic cells, playing a critical bridge role between innate and adaptive immunity. It activates NK cells, enhancing their cytotoxicity. Crucially, IL-12 drives differentiation of naive T cells into Th1 cells, promoting cell-mediated immunity. IL-6 is produced by macrophages and T cells with multiple targets. It promotes B-cell differentiation and antibody production. It also enhances T-cell activation, growth, and differentiation. IL-6 is another acute phase reactant inducer. TNF-alpha is produced by T cells and macrophages, named for its ability to cause tumor necrosis. It increases cytotoxicity of cytotoxic T lymphocytes, or CTLs. TNF-alpha also activates endothelium, promoting leukocyte adhesion and extravasation. However, excessive TNF-alpha causes septic shock, explaining why anti-TNF therapies help in certain diseases. IFN-alpha and beta are produced by macrophages and fibroblasts, providing immediate antiviral defense. They increase MHC class I expression, enhancing antigen presentation. They also increase NK cell activity, promoting elimination of infected cells. These cytokines collectively initiate protective inflammatory responses. Cytokines of adaptive immunity are produced primarily by activated T cells, fine-tuning immune responses based on pathogen characteristics. Understanding these is essential for grasping how immune responses are coordinated. IL-2 is produced by Th1 cells and is the major T cell growth factor. It stimulates proliferation of activated T cells through autocrine and paracrine mechanisms, causing clonal expansion. IL-2 also activates NK cells, enhancing their cytotoxic function. Clinically, IL-2 is used in cancer immunotherapy. IL-4 is produced by Th2 cells and is the defining Th2 cytokine. It promotes B-cell proliferation and differentiation. Crucially, IL-4 drives class switching to IgG1 and IgE antibodies, IgE being particularly important in allergic responses and helminth infections. IL-4 increases MHC class II expression on B cells and promotes T cell proliferation, creating positive feedback for Th2 responses. IL-5 is also produced by Th2 cells, stimulating B cell growth and increasing immunoglobulin secretion. Importantly, IL-5 activates eosinophils, which are crucial for fighting helminth parasites. Elevated IL-5 is associated with allergic diseases and asthma. IL-5 is produced by Th1 cells and is the signature Th1 cytokine. It inhibits viral protein synthesis in infected cells, providing antiviral protection. IL-5 powerfully activates macrophages, enhancing their microbicidal activity critical for fighting intracellular pathogens like mycobacteria. It stimulates IL-1 and IL-2 production and promotes antibody production, particularly IgG subtypes effective in opsonization. The balance between Th1 and Th2 cytokines determines the character of immune responses. Cytokines orchestrate immune responses. Understanding these roles reveals how cytokines coordinate the complex cellular interactions required for effective immunity. First, cytokines mediate activation of immune cells. Resting lymphocytes and macrophages require cytokine signals to become fully activated. For example, IL-2 activates T cells, while IFN-gamma activates macrophages. This ensures cells are activated only when needed. Second, cytokines control differentiation of immune cells into specific functional subsets. IL-12 drives naive T cells toward Th1 fate, while IL-4 drives them toward Th2 fate. This differentiation determines whether responses will be cellular or humoral. Similarly, cytokines influence B-cell class switching, determining which antibody isotypes are produced. Third, cytokines guide maturation of immune cells, promoting their development from precursors into fully functional effector cells. Colony-stimulating factors like GCSF and GM-CSF promote development of granulocytes and macrophages from bone marrow precursors. Fourth, cytokines provide regulation of immune cells, both amplifying and limiting responses. Some cytokines like IL-2 amplify responses through positive feedback. Others like IL-10 and TGF-beta suppress responses, preventing excessive inflammation. This regulatory function prevents immunopathology, where immune responses cause more damage than the pathogen itself. Through these mechanisms, cytokines create a coordinated network of cellular interactions that efficiently eliminate pathogens while minimizing collateral tissue damage. Dysregulation of cytokine networks contributes to immunodeficiency, autoimmunity, and inflammatory diseases. These examples illustrate how basic immunology translates into clinical treatments. First, interferons are used in treating viral diseases and cancer. IFN-alpha treats chronic hepatitis B and C infections, inducing antiviral states in infected hepatocytes and enhancing immune clearance of virus. IFN-alpha also treats certain cancers, including hairy cell leukemia and melanoma, by enhancing immune surveillance and directly inhibiting tumor cell proliferation. Second, several cytokines enhance T-cell activation in immunodeficiency diseases. IL-2 administered therapeutically can boost T-cell responses in some immunodeficiencies. IFN-gamma treats chronic granulomatous disease, where phagocytes can't kill ingested bacteria. TNF-alpha has experimental uses in enhancing anti-tumor immunity. Lymphokine-activated killer cells, or LAK cells, generated by culturing patient lymphocytes with IL-2 ex vivo, are used in cancer treatment to boost anti-tumor responses. Third, GM-CSF, or granulocyte macrophage colony stimulating factor, induces increases in white blood cell counts by stimulating bone marrow production. It's used to restore leukocyte counts after cytotoxic chemotherapy-induced neutropenia, which otherwise leaves patients vulnerable to life-threatening infections. GM-CSF is also used after bone marrow transplantation to accelerate engraftment and restore immune function, and to correct AIDS-associated leukopenia, where HIV causes immune suppression. Fourth, GCSF, or granulocyte colony stimulating factor, specifically increases neutrophil counts and is used in neutrophil deficiency conditions. These applications demonstrate cytokines' therapeutic potential. Beyond using cytokines therapeutically, blocking cytokines has proven highly effective in treating immune-mediated diseases. This represents a major advance in modern medicine. Anti-cytokine antibodies are used in managing autoimmune diseases and transplant rejection. The principle is blocking pathogenic cytokines driving disease processes. Anti-TNF therapy has revolutionized treatment of rheumatoid arthritis. TNF-alpha is a key inflammatory cytokine in RA, promoting joint inflammation and destruction. Biologic drugs like infliximab, adalimumab, and etanercept block TNF-alpha, dramatically reducing inflammation and preventing joint damage. Anti-TNF therapy also treats inflammatory bowel disease, including Crohn's disease and ulcerative colitis, psoriasis, and ankylosing spondylitis. Anti-IL-2 receptor antibodies reduce graft rejection in organ transplantation. Basiliximab and daclizumab are monoclonal antibodies blocking the IL-2 receptor on activated T cells. Since IL-2 drives T-cell proliferation and graft rejection, blocking this pathway suppresses rejection while being more specific than traditional immunosuppressants. Other cytokine blocking therapies include anti-IL-1 for autoinflammatory syndromes, anti-IL-6 for rheumatoid arthritis and Castleman disease, anti-IL-5 for severe asthma with eosinophilia, and anti-IL-17 for psoriasis. These targeted biologic therapies are more specific than traditional immunosuppressants, providing effective disease control with fewer side effects. However, they increase infection risk since they suppress immune responses. Understanding cytokine biology enables rational design of such therapies, exemplifying how basic immunology translates into clinical breakthroughs.